Okay, welcome everyone. Welcome once again to developmental biology. Can you put your faces up real quick just so we can pretend we're a class here? Hey, Elijah. You're a good looking man, Elijah. Don't let anybody tell you different.
Thank you.
Hannah Rondo, Josh Wolford, Vanessa. I give everyone permission to call Vanessa Gordon Nessa if you want to. She likes that. Akiola Simon, yay. Kwame, are you still a student? I thought you dropped out. He's my friend, Kwame. We got 23. Going in a second here. So I got good news for everybody. Today I got to the end of what I put on the first exam last year. So we get to learn more stuff before the next exam because we still have a lot of time. So a lot of chapter 3 I didn't cover last time. I'm going to go over now, maybe starting today, but definitely Friday.
Good question. Does that mean that what we cover now will be on the exam, or we're just going ahead of schedule?
Everything before the exam will be on the exam.
Okay.
I like the Zoom stuff. I get to see myself. You know what's his name? The rapper who married Kim Kardashian. What's his name?
Kanye West.
Kanye West said his greatest disappointment in life is that he'll never get to see himself perform. So I get to see myself perform now, which is a good thing, I guess. Bill Barr has a whole spiel about Kanye West. It's kind of funny. But anyway, I'm not going to go into that here. All right, well, let's go ahead and get started. See what the heck we came here for. Here, my friends, is my PowerPoint. Mm-hmm. People are still filtering in. It's okay, they're allowed. Still allowed in. Just arranging here because I want to look at my notes at the same time to make sure I don't forget anything. I did upload my notes and the PowerPoint. I added a whole bunch of slides to it, so that's new too. Okay, human development. In cell biology the other day, we should probably watch this video we watched in cell biology. I've been covering microtubules, and they're in cilia, and cilia are involved in the fallopian tube. So I showed that a video yesterday on that. Let's watch that just to ease into our discussion today. Let's see if I can find it real quick. So I know most of you are unfamiliar with how fertilization occurs in the human. Is it? I don't know if you're familiar with the whole process of pregnancy. I know you're millennials and you come from a very sheltered background. So motility. Okay, here we go. two short videos here. One is ovulation. Oops. Hang on. Stop. One is ovulation, and the other is coming from the other end, the male version, which is Mr. Sperm swimming up the tract. Okay, here we go.
Ovulation is a part of the menstrual cycle when the ovary releases a ripe egg or ovum. Inside the ovary are hundreds of thousands of follicles. Each follicle is a hollow ball of cells with an immature egg in the center. The typical 28-day menstrual cycle begins on the first day of menstrual bleeding. During the first 7 days of the cycle, a few follicles begin to grow at the same time. These maturing follicles secrete estrogen hormone into the blood.
Professor, I can't hear it that well. The sound is very, very low.
Yeah, there's not much I can do about it. I've got my sound up all the way. Maybe you could turn your sound up all the. Oh, wait. Maybe I can get this a little louder. I know it doesn't. It never comes out well when I do YouTube on this, but.
Around day 7, all of the follicles stop growing and begin to degenerate, except for one. This dominant follicle continues to grow and nourishes the developing egg inside it. Around day 12, the follicle secretes a large amount of estrogen into the bloodstream. When the estrogen reaches the hypothalamus and the pituitary gland in the brain, the anterior or front part of the pituitary gland releases a huge surge of luteinizing hormone into the bloodstream. Around day 14, luteinizing hormone causes the follicle to undergo a sudden growth spurt. Right before ovulation, the egg detaches from the inside of the follicle. The bulging follicle releases chemicals, causing one of the two fallopian tubes to move in closer and surround the follicle. The follicle swells until it bursts open, ejecting the egg and fluid from the follicle into the abdominal cavity. In response, the fimbriae, tiny projections at the end of the fallopian tube, sweep across the ovulation site and pick up the egg. Microscopic cilia on the fimbriae surface transport the egg to the entrance of the fallopian tube. Inside the walls of the fallopian tube, muscular contractions gently push the egg towards the uterus. After ovulation, the egg lives for 12 to 24 hours, so it must be fertilized by a sperm from the male during this time for a woman to become pregnant. If it's not fertilized, the egg dissolves away and is shed along the uterine lining during menstruation.
Now we're going to attack it from the other side.
Fertilization is the epic story of a single sperm facing incredible odds to unite with an egg and form a new human life.
It's truly a miracle that happens 10 million times a day.
The story of all of us. During sexual intercourse, about 300 million sperm enter the vagina. Soon afterward, millions of them will either flow out of the vagina or die in its acidic environment. However, many survive because of the protective elements provided in the fluid surrounding them. Next, the sperm must pass through the cervix, opening into the uterus. Usually, it remains tightly closed, but here the cervix is open for a few days while the woman ovulates. The sperm swim through the cervical mucus, which is thinned to a more watery consistency for easier passage. Once inside the cervix, the sperm continues swimming toward the uterus, though millions will die trying to make it through the mucus. Some sperm remain behind, caught in the folds of the cervix, but they may later continue the journey as a backup to the first group. Inside the uterus, muscular uterine contractions assist the sperm on their journey toward the egg. However, resident cells of the woman's immune system mistake the sperm for foreign invaders, destroying thousands more.
Yikes.
Next, half the sperm is for the empty fallopian tube, while the other half
Suckers.
is only 3,000 remain. Inside the fallopian tube, tiny cilia push the egg toward the uterus. To continue, the sperm must surge against this motion to reach the egg. Some sperm get trapped in the cilia and die. During this part of the journey, chemicals in the reproductive tract cause the membranes covering the heads of the sperm to change. As a result, the sperm become hyperactive, swimming harder and faster toward their destination. At last, the sperm reach the egg. Only a few dozen of the original 300 million sperm remain. The egg is covered with a layer of cells called the radiata. The sperm must push through this layer to reach the outer layer of the egg, the zona pellucida. When sperm reach the zona pellucida, they attach to specialized sperm receptors on the surface, which triggers their macrosomes to release digestive enzymes, enabling the sperm to burrow into the layer. Inside the zona pellucida is a narrow, fluid-filled space just outside the egg cell membrane. The first sperm to make contact will fertilize the egg. After a perilous journey and against incredible odds, a single sperm attaches to the egg cell membrane. Within a few minutes, the outer membranes fuse and the egg pulls the sperm inside. This event causes changes in the egg membrane that prevent other sperm from attaching to it. Next, the egg releases chemicals that push other sperm away from the egg and create an impenetrable fertilization membrane. As the reaction spreads outward, the zona pellucida hardens, trapping any sperm unlucky enough to be caught inside. Outside the egg, sperm are no longer able to attach to the zona pellucida. Meanwhile, inside the egg, the tightly packed layer of genetic material spreads out. A new membrane forms around the genetic material, creating the male pronucleus. Inside, the genetic material reforms into 23 chromosomes. The female genetic material, awakened by the fusion of the sperm and the egg, finishes dividing, resulting in the female pronucleus, which also contains 23 chromosomes. As the male and female pronuclei form, spiderweb-like threads called microtubules pull them toward each other. The two sets of chromosomes join together, completing the process of fertilization. At this moment, a unique genetic code arises, instantly determining gender, hair color, eye color, and hundreds of other characteristics. This new single cell, the zygote, is the beginning of a new human being. And now the cilia in the fallopian tube gently sweep the zygote toward the uterus, where he or she is planted in the rich uterine lining, growing and maturing for the next 9 months until ready for birth.
Okay, so that's everything that happens before we take off on our journey. Oh, come on. Stop share. Okay. So we're going to take up the story right at that point where the egg has been fertilized and now we've got a zygote. And then as the fertilized egg, then the zygote goes down the fallopian tube towards the uterus, it continues to divide. So it'll get as far as the blastocele stage before it gets to the uterus, which is important. Okay, so here, oops, shoot. Damn it hell. I'm still not good with my multimedia use here. Okay, so here is your egg. Egg meets sperm. And now here it divides into 2, 4, 8. So we're getting all the way to the blastula stage, which looks very similar to the chick blastula and the mouse even. So we've got inner mast cells and then trophectoderm around it. So that is the beginning. Okay, so now most of what we talk about, I'll talk a little bit about gastrulation and neurulation here, but it's pretty much the same as the chick. I mean, it's different from the mouse because it's not in that cylinder shape and you don't have the turning that happens with mouse. But other than that, you know, it's all pretty much the same. What's really interesting in this part of the chapter is they go in detail into how the placenta forms and the umbilical attaching to it. So here at the blastula stage, when it first starts to implant, you've got epiblast cells here. That's the actual embryo itself. And then underneath it, you've got hypoblast cells. So these are bilaminar. Okay, so you got 2 layers of cells. The top are ectoderm, basically. They're ectodermal cells. And then you've got hypoblast cells circling the yolk here. And outside of that, you've got extraembryonic mesoderm, and these down here will be your trophoblasts. Okay, so here we are again. Now we're at the gastrula stage. Let me see if I forget something. Oh, sec. So the stalk here is extraembryonic mesoderm. That will become the umbilical cord. So our placenta is going to form out here and the umbilical cord will attach to that. Okay, so gastrulation. Primitive streak starts at the posterior end and goes forward. So again, like the chick, it's like a flat disc of cells, and that primitive streak goes forward, and epidermal cells here, ectoderm, will divide and converge and extend. So they'll go in through that primitive streak and go forward and laterally. And then because of that, you end up with 3 layers of cells. Ectoderm, the cells that stay on top are ectoderm, mesoderm, and endoderm get specified as they go through. So they change cell type as they go through the primitive streak. They become mesoderm and endoderm. Endoderm then goes on top of that hypoblast cells, or hypoblast cells. So then you end up with endoderm, mesoderm, and ectoderm, 3 layers of cells. So very similar to the chick at this point. Mesoderm, and then after this, once you get this going up, then neurulation happens in the opposite direction. You get mesoderm being laid down as notochord, and that will induce your neural plate. The neural plate then starts to fold up, so you get neural folds, and then it closes up to give you a neurotube. Okay, getting a little bit repetitive. We saw this with several other examples. And then once that happens, once it's neurulated, it will, let's see. Once you've gone through neurulation, here's your neural plate on top, notochord here, somites on either side, and endoderm. You've got your 3 layers there, ecto, meso, and endo. And then neurulation starts to happen with the groove. And so here your neural tube is all sealed up, epidermis all over the whole thing, somites at either side. And you can see down the bottom, the endoderm has surrounded the yolk, and then it closes up underneath. Let's see that here. This is another series from your book that I think gives you a better view of the way the body cavity closes up with the endoderm on the inside. So here we're at that 3-cell stage, or 3-layer stage. You've neurulated here. You've got the neural fold, and then you've got red mesoderm in the middle, endoderm here. The top and the anterior and posterior sides start to bend down in, And see, they're bending down in here and here. And then, oh, come on. And then you can't really, it's hard to see here because it's in 3 dimensions. Those sides are folding in. You've got ectoderm folding in from the outside, covering the body cavity. And on the inside, the endoderm is also folding in, forming the gut. Let me do that again. Okay, so you've got 3 layers here, and then that ectoderm is going to keep expanding and doing this, kind of like epibole, where it's going out and around. Top and bottom are coming in, and the endoderm closes up as the ectoderm comes around it and seals the whole thing up. And the only place it doesn't seal is here, and this is going to be where your umbilical cord comes out.
Is the neural tube and the brain like at the same place?
The neural tube is more like the spinal cord. The brain happens anterior to the neural tube.
Okay, thanks.
Sure. If you remember when neurulation happens, right before the node recedes, mesoderm comes in and through and goes forward and forms the head fold. And that's the mesoderm that's going to induce the brain to form. Okay, so that's the folding up and stuff. That's all well and good. Not, like I say, not that different from the other models we saw. So here, this is kind of the same as mouse, but they didn't go into it when we did the mouse. So we'll talk about the formation of the placenta more here. So here with the placenta, hang on a second. Okay, so here is your blastocyst. Okay, so you've got the deep mast cells here, and in yellow are the trophectoderm cells around it. And those are extra embryonic, and a fluid-filled blastocele here in the center. Now these pink cells here are the maternal uterine cells. And right here, the trophectoderm here start to fuse together and form giant cells like we saw in the mice, and they become a big syncytial type of cell, and they invade inside the uterine cavity. So this is like, now you flipped over. Okay, so here's your blastocele back here. Here's the epiblast here, and this is the hypoblast there. And then here, these green things, these multinucleated green things, are the giant cells, and they're called syncytiotrophoblasts. Syncytiotrophoblasts. So it sounds like a complicated word, but we've seen this before where you get cells fusing and forming multinucleated cells. That's a syncytium. So this is syncytiotrophoblasts. So these are syncytial tissue that comes from the trophoblast. The other extra embryonic cells that are important here are the cytotrophoblasts. So these are trophoblasts that remain individual cells. Okay, so you start out with trophoblast cells, and the trophoblast cells split in 2 different ways. Some became syncytial, some stayed cells. So you get syncytiotrophoblasts and cytotrophoblast cells. The cytotrophoblast cells surround the whole embryo, and they form that barrier that I believe becomes the amnion or the chorion. I forget now, one or the other. But the syncytials then will invade into that uterine wall. See what you put your mother through. Just watch this. It's horrible. Okay, so here we are again. Here's your blastula, blastocyst coming down. It hits the uterine wall, attaches. You form these giant cells, the syncytial trophoblasts, and then the whole thing invades and the uterine wall grows over it. And the embryo will always be covered by the cytotrophoblasts. Okay, once this happens, once these syncytiotrophoblasts have invaded, okay, they form this thick layer of syncytium around the embryo, and it starts to give off HCG, human chorionic gonadotropin. We all know what that is, right? That's how you know when you're pregnant, when your body gives off so much HCG, it ends up in the urine, and then you can test for it in the urine. Takes about a week or two. I think this implantation happens around day 8, I think, or something. So after that is when you can start to detect it in the urine. So this HCG is really important. It's actually structurally similar to the luteinizing hormone that we saw earlier involved in follicle maturation. So the HCG then will induce the ovaries to start releasing more progesterone. So you're going to get a progesterone spike here. And that progesterone then causes the uterus to develop more vasculature. So the vasculature starts to develop here as a response to the HCG. So you can see the embryo and the mother talking to each other. The embryo is giving off HCG and the mother's producing progesterone. And together they're developing that uterine wall to help the implantation happen. Okay, so the embryo is embedded and surrounded by a single layer of cells, the cytotrophoblasts. And then you've got this thick syncytial layer here, and that's going to be really important here, we'll see. So now inside the syncytial layer, you can see these little circles here. These are vacuoles that start to form. And these vacuoles then will fuse with maternal capillaries. And this is where you start to make this placental apparatus here. So in the placental apparatus, you've got all of these structures here, like 1, 2, 3, 4 of these structures sticking out, and they are called villi. So we can look at one villus up close here. So this is one villus. So look at the structure here. This is really freaky. This really blew me away the first time I saw it. So these little cells here are your cytotrophoblasts, and everything embryonic is covered by that. So inside is the embryo, outside is not really anymore. Okay. Now this blue is that syncytial layer, the syncytiotrophoblasts. So you've got a bit of space there with the multinucleated space. Outside of that, oops, sorry. Let me blow this up. Okay, so you've got the cytotrophoblasts surrounding everything. You've got your syncytial layer surrounding the cytotrophoblasts, and then you've got this open space here. And this open space is the intervillus space, right, between the villi, intervillus. And the uterine artery just dumps blood out into there. So, you know, we always talk about the human circulatory system being closed. Here it's practically open because the maternal uterus is just dumping blood into this intravillus space. And that surrounds these villi. So then you're getting blood and oxygen, carbon dioxide, nutrient waste exchange across this trophoblast layer. And inside that trophoblast layer are the umbilical, the umbilical vein and the umbilical artery. So artery is bringing oxygenated blood back. Is that right? No, arteries go away from the heart. So I guess the artery is coming from the embryo and the vein is going back into the embryo. And then this blood in this empty space here, or open space here, just eventually makes its way back into the uterine vein and gets sucked back into the maternal body. So you can see here, like in my other class of pharmacology of recreational drugs, I always talk about, if a woman is pregnant, the fetus sees the same drugs that the mother's taken, because there's really very little separation here between the mother and the fetus. So anything in the maternal blood will end up in the fetal blood as well. I believe antibodies can also cross that. I'm not really sure what they do, because if the mother gets infected with something, the baby can get immune to it as well as the mother. So this is kind of cool. Here's your maternal arteriole dumping blood out into that intervillous space, and it ends up coming back into the veins and other places, and then back up into the mother's body. Okay. So what did we learn here? New words are the villi and the villus space, the umbilical vein and umbilical artery, syncytio trophoblast, one of my favorite words. You know what? If you're ever in a spelling contest, use that word as your challenge to your opponents. Spell syncytio trophoblast. Use it in a sentence. Okay, any questions? Mairead, you're leaning forward there. Did you have a question?
I kind of did. It's more of like an inquiry than a question.
Mm-hmm.
On the last slide, there was extravillous trophocyte layers that are along the past uterine wall, and I wanted to know what that was about.
That's a good point. It's a point that I was hoping no one would ask because I didn't put it in my notes. Let's look back at the book because it does. Oops. Shoot. It does talk about those. Oh, come on, man. They're really important because they help to keep the placenta. They keep the placenta working. Hang on already. Come on, come on, come on. There it is. Okay, so trophoblasts produce differentiate syncytio. The extravillous trophoblast cells migrate into the uterine wall, helping to anchor the placenta and remodel the uterine arteries, which increases maternal blood flow into the intravillous spaces. These functions of the extravillous trophoblast cells are crucial for the establishment of a fully functioning placenta. Defective placental development can lead to serious clinical conditions, including preeclampsia, intrauterine growth retardation, miscarriage, and stillbirth. Okay, so they're helping to keep building more blood vessels, probably releasing HCG and progesterone, causing progesterone release. And that's going to keep a good mother-fetus relationship going. I guess you can't see what I'm doing. Here. Okay, so it's right here, page 135 of your book. Extravillous trophoblasts migrate. Oh, shoot. Okay. Otros problemas, otras preguntas? Any other questions? Because I'm going to go on and talk about experimental techniques next. And it's kind of weird in your book because they went through all the different models of the mouse, then they did experimental techniques, and then they did human stuff. So I skipped all the techniques. Now I'm going back and I'm going to go through the techniques. So let us do that. Hey, Rolla. Okay, so as you maybe would be able to imagine, a lot of the early work and the stuff that we've talked about already. Oh, shoot, I didn't talk about this. Let's talk about twinning really quick. This is the last thing in human development that they talk about. Is anybody out there a twin, identical twin? Can someone say yay on the chat or something? Let's stop share. Anyone an identical twin? Nobody? Okay. Well, I didn't know this until I read this. There are 3 different ways to get identical twins. One, what I always assume was the way you get a twin, is when you have a zygote at the 2-cell stage, those 2 cells split. So you end up with 2 separate cells instead of 1 2-cell embryo. And then each of those 2 embryos or 2 cells develops in a blastocyst, which then implants and forms 2 different fetuses. So let me just continue and I'll come back. Another way is you get to the blastocyst stage, and then that splits into 2 embryos, and then that implants. The third way is the whole thing. You've got a single blastocyst that implants, and then it splits into 2 embryos. And you can see the difference here that you can see is here. The pink is the chorion, and the yellow is the amnion. The outside membrane of the amniotic fluid here is the amnion. So here in this type of twinning, you have each fetus has got its own umbilical cord, its own chorion, and its own amnion. Here in this type of twinning, you end up with one amnion, which is the pink, surrounds them all. They both have their own umbilical cord, and they each have their own amnion, an amniotic sac. And in the third type, they share the chorion. They each have their own umbilical cord, but they're in the same amniotic sac. Okay, so 3 different ways that can happen. This isn't that important. I don't think I would ask you this on a test, but it's interesting. Here's something. If you have a very strong stomach, do a Google Images search for fetus in fetu. Fetus in fetu. That means when there's one fetus inside the other one, which I imagine happens because of some mistake in this process here. And that's, you've probably seen this where someone's got like legs sticking out of their chest because another fetus is coming out, another individual is coming out of this individual. A lot of it happens at the mouth where like the baby is born and there's like a whole mass coming out of its mouth that's actually another fetus coming that's attached by the mouth to this fetus. Pretty weird stuff. If you've got a strong stomach, do that Google Images search. If you don't, then I suggest you don't. Not for the faint of heart. Okay. So moving on to molecular techniques. Oh, shit. Then there's this slide I forgot to talk about. So you can see where the different tissues come from. So you start out with your blastocyst. Now at that blastocyst stage, you've got trophoblast cells and inner mass cells. We can see that here, trophoblast around and inner mass in the middle. Those trophoblast cells will then go on to become cytoblasts or syncytioblasts, and that helps to develop the placenta. Of the inner mast cells, you've got hypoblast, which is extra embryonic, and it becomes the extra embryonic endoderm, which becomes yolk sac. And the epiblast, most of which is going to be the embryo, but some of it becomes the amniotic ectoderm, so the membrane of the amnion. And the rest of the epiblast then, it starts out as pretty much all ectoderm. Some of that ectoderm then goes through the primitive streak and becomes endoderm, and some goes through the primitive streak and becomes mesoderm. So that's kind of where it all starts and where each of those tissues ends up. And they figured these things out through a process called fate mapping, which we'll talk about on Thursday. One really cool way to do fate mapping is here, you can go to this inner mast cell, right? Take a needle, poke into the embryo, and inject a non-diffusible dye into one of the cells of that inner mast cell, and then let the embryo develop and find out where those colors go. And you'll see where each of those cells ends up in the final embryo. So we'll talk about that technique on Friday. Today, we're going to talk about other techniques, though. So again, I started saying this before I forgot about those last two slides. So old school, we're talking 19th century till probably the mid 20th century. How do they do this embryology? Basically, careful observation and detailed drawings. They weren't really able to take pictures of things that were really small like that. The technology wasn't there. But so they would, just look at things really closely, take detailed drawings of it, drawing detailed pictures, okay. Dissection, right? Cutting them apart and see what pieces are what. And after you've done this dissection, you could take pieces from one embryo and add them to a different embryo. So that's what I'm talking about here with transferring blocks of tissue. So in all of these models we talked about, one thing that keeps coming up is the node. And I said the node's really important. What you can do is take the node out of one embryo and put it into another embryo and see what that does. So then you can figure out what the node does, because the node is giving off a lot of hormones, we'll find out in the next chapter, and that's changing cell types and making cells different. So that's another kind of classic technique that people did to find out what's going on in the embryo. And then, like I just talked about, injecting non-diffusible dyes, poking an embryonic cell and putting a non-diffusible dye into it. And then as that cell divides, every cell that came from that cell will have the dye in it, and you can see where those cells end up. So that's the classical techniques. More modern techniques come in once molecular tools are available. And now we can do just about any, with CRISPR, you can do anything you want genetically. You can mutate a gene, you can knock it out, you can mutate a gene and make it active or inactive. You can express it so you've got more. You can turn it on at a time when it's not supposed to be turned on. You can do anything you want to a particular gene. So if you want to know what a gene does in embryonic development, how is it important, you can just monkey with the gene and see what happens when you change it. Another thing another two techniques that are really powerful are fluorescent antibody detection and nucleotide labeling. So these are two techniques I'll talk about in a second that are all about finding out when genes are turned on, finding out where those genes are turned on and what proteins are being made where and when, which tells us a lot about what's causing these embryos to develop. And then finally, imaging. You can get really nice, clear pictures through an embryo because each of the tissues is a slightly different density, so you can see, different tissues developing throughout the process. Okay, so we'll start out with in situ nucleic acid hybridization. So in situ means in the situation, like in the embryo itself. So you take an embryo and you fix it. You make it still, probably, I think you kill it too at this point. And then you take a labeled DNA probe. So say there's a specific gene you care about. When is the gene for activin turned on? As soon as the gene's turned on, the messenger RNA will show up. So you take a DNA probe against that messenger RNA, and you probe the embryo with it. Now, technically, how do they do it? I don't know. I've never done it, but I imagine they take the embryo, they flatten it out, and stick it to a paper, and then they stick their DNA probe in there, and then that DNA probe will have some kind of fluorescent tag on it, and you can get different colors of fluorescent tags. So you can do different DNA probes with different colors showing up. And so by taking embryos at different stages and doing this experiment, you can see what tissues this particular gene is being turned on in, and when during the period of development, the genes are being turned on. This is from your book, chapter 1, actually. So you're taking your gene of interest is A. When is A going to be made? These are messenger RNAs from the embryo. So the embryo makes a lot of different messenger RNAs. So you take your thing, you expose the embryo to it, and your probe is going to stick to only the genes that you're interested in, so you can see where it is. And this little star next to the A says that it's fluorescent, so you can see it. This is the kind of data you can get from that. Isn't this beautiful? So here, this is a neurula. So this is a chick that's at the neurula stage, and they're looking for this gene TBX6L. I think it's a transcription factor. And you can see this is being expressed in neural tissue, heavily here in the neural tube and a little bit more in the neural folds on either side. Here's Claudin-1, which is expressed up here. Oops, shoot, not what I wanted to do. Okay, so Claudin-1 is expressed heavily out here. You can see this TBXL1 is not expressed up here, but the Claudin-1 is. Here's GATA4, and it's being expressed here and nowhere else. Okay, so again, trying to find out what genes are being turned on in what tissue, and that helps you understand why those tissues are differentiating in the way that they are. And we'll get into all of that in the next few chapters, really the rest of the class. Here's a really cool picture. So this is 1, 2, 3 different probes. You've got your red, your green, and your yellow. So 3 different DNA probes, SOX2, SOX10, and GATA2. These are all transcription factors. And you can see where each of them lights up. I think the yellow is the overlap of green and red. And blue is GATA2, which is everywhere else. Okay, so that's one really neat, powerful tool. Another one is, oh, here's 4 different probes. Look at this, isn't this amazing? So here's what the embryo looks like under the light microscope. This is one particular gene that they're looking at, and it comes up and they have a red fluorophore on it. Here's a different gene with a green fluorophore. Here's one with a blue fluorophore, and here's one with a purple fluorophore. So in each of these little pictures, you can see exactly where those genes are lighting up. What tissues are they being expressed in? And here you can overlap them all and see kind of an overall view of where these things are being turned on. The neat thing about a fluorophore is it'll be excited at a specific wavelength. So you can only excite one fluorophore and see only where that gene is. And then go to different wavelengths to see different genes. Or you can turn all, oops, shit, sorry. Turn it all on at once and you end up seeing it all. Okay. All right. Another technique, we're almost done here. I think I let you out early last time, so I'm going to keep you a couple minutes late here today. Another technique is immunohistochemistry. So basically, you've got a protein you're interested in, you make an antibody against it, and then you add a fluorophore to that antibody. So, you know, this is a protein, maybe it's a hormone that comes up at some point during the process. You want to find out where and when. You make an antibody against it, you add a fluorophore, and you stain your embryo with it. Another thing you can do is you can make your antibody and get a different antibody that will bind to it. So, for instance, you may make your antibody, usually a monoclonal antibody, against the protein you're interested in. maybe you make that antibody in the mouse. Usually that's where you make monoclonal antibodies. And then rather than adding a fluorophore to your antibody, you just go out and buy goat anti-mouse antibodies. So these goat antibodies will be directed against the constant areas of the mouse monoclonal antibody, and they already have a fluorophore attached. That's the neat thing about molecular biology today. You don't have to do all this stuff. You can probably buy your primary antibody, and you can definitely buy these secondary antibodies just to make the work go faster. And so here's an example of that. Here is sonic hedgehog, which is a hormone that's really important in neural development. And you can see it lights up all the way down the neural tube. And these other ones, they've taken this and like sliced through the embryo at different stages, like through the head. You can see hedgehog is in the neural plate, as well as the notochord. Come down a little further at the level of the first somite, you've got a lot less hedgehog in the neural plate, a lot in the notochord. Come down to the middle, there's hardly any left in the neural plate, but it's still in the notochord. Come down to the bottom, it's only in the notochord. So you can see exactly where that protein is being produced. And here's just multiple antibodies, just to show you that you can do the same thing here that you did with the other one, where you've got multiple different antibodies. You know, I'm looking at this, it looks 3D to me. My eyes are getting weird. All right, so that's about it that I have for you today. And we've run over by a couple minutes, but I think that's okay. Any questions or comments before we say goodbye? Okay, guys, I'll see you again on Friday. Have a great rest of your week.